All right, now chapter 5, the last chapter of the first unit. This is all about large biological molecules, basically carbohydrates, lipids, nucleic acids, and proteins. These large organic molecules that are the basic, or the building blocks of living things. Um, but first, we refer to these large molecules. Three of the four are classified as polymers. Um, the lipids are not technically, but polymers. Polymers are large molecules that are just these basic subunits bonded together one after another, and they can be extremely large, consisting of thousands of these subunits. Um, when we put them together or take them apart, what we do, so when we put them together, we do what's called a dehydration reaction, because what's happening is you're removing the hydrogen of one of those subunits, the hydroxyl of the other, generating some water, and bonding those two subunits together. So, excuse me, so essentially water is being given off, dehydration. Well, to break them apart, you do the opposite. You do a hydrolysis. We're going to split, lyse means to split, with water. We're going to add water and break apart that polymer. So, dehydration, synthesis, and hydrolysis reactions are the way we put together and take apart these polymers. All right, first, carbohydrates or sugars. Okay, so we have simple sugars known as monosaccharides, and here are some simple sugars: glucose, galactose, fructose. Um, and if I'm not mistaken, if you look at these, they are all isomers of each other, all same chemical formula, but a different arrangement of the molecules. You can see the fructose has this carbonyl group towards the inside of the molecule, whereas glucose and galactose have it on the end. Um, so a ketone here, or ketose, and an aldehyde or an aldose in the case of these two. Um, Monosaccharides. Glucose is going to be the one we're going to revisit quite a bit. All right, now when you take several of these monosaccharides and put them together, you get a polysaccharide. So essentially a polysaccharide is the sugar polymer. So here we're going to do our um, dehydration reaction. We're going to remove the water. We're going to bond these two glucose together, forming technically here what's called a disaccharide or maltose sugar. Um, and here we take a glucose and a fructose and put them together, and we get a disaccharide known as sucrose, which is basic table sugar. But when we put many more of them together, we get a true polysaccharide or sugar polymer. Starch and cellulose are some good examples. Now, notice that here's starch on the left, cellulose on the right. They are both polymers of glucose molecules, but they use slightly different forms of the glucose in putting them together. So we could describe starch and cellulose as isomers. Essentially, they're made of the same thing, but with a somewhat different arrangement. You can see with starch, the hydroxyl groups are all on one side, and with cellulose, they alternate between sides. Um, but again, just long strands of glucose. Starch um, is what you find in plants, potatoes, for example, um, as the storage. It's how they store energy. Cellulose is the polysaccharide they use to build cell walls. So it's what you might call a structural sugar um, with cellulose, and a storage sugar with starch. Um, Animals, it's not really pictured here, have a form of a sugar polymer called glycogen, and it's how we store our carbohydrates, particularly in the liver. Um, Now here's another one, chitin. So chitin is a polymer of a modified glucose that has this structure stuck onto it, and excuse me, it's what it's used to make the exoskeleton of insects. Um, okay, lipids. So um, lipids are these are the ones that are not technically polymers because they don't they're not just hundreds or thousands of repeating subunits. They have a set structure, but they are large molecules and very important ones. So you see you have this glycerol here, which is a type of simple 3-carbon sugar. And what you're doing is a dehydration reaction, and you're bonding this long chain of primarily carbons and hydrogens, known as a fatty acid, onto it. And when you put 3 of these on there, you essentially get your basic um, lipid, or in this case, what's called a triacylglycerol. 3 fatty acids and 1 glycerol. Now notice the fatty acids, this one's slightly shorter, but that's not a big deal. This is also known as a fat. Fats are lipids that have fatty acids that are straight and have no double bonds between the carbons. Another type of lipid are the oils, and oils still have the glycerol, the 3 fatty acids, but at least one or sometimes more of their fatty acids have a double bond between the carbon, one of the carbons, which causes the chain to bend. And by causing it to bend, what that means is the fatty acids don't pack in very closely, and so they are basically liquids at room temperature, whereas fats are solids. And again, more than one of those um, fatty acids can have the double bond. These are known as unsaturated fats because they are essentially, to make that double bond, you have to remove some hydrogen, so they're not saturated with hydrogens. Whereas in fats, the fatty acids are known as being saturated. And you probably know this from nutrition and nutrition labels on foods and stuff like that. Generally, the saturated fats are considered somewhat unhealthier because they tend to clog up your arteries. All right, another type of lipid, phospholipids. Excuse me. These are lipids that have the glycerol. They have 2 fatty acids, and they can have the straight chain or bent chain. They have a phosphate group, thus phospholipid, and this other molecule called choline. And phospholipids, as you may recall from freshman year, are the primary component of biological membranes. They form a 2-layer structure known as a phospholipid bilayer, with the phosphate choline end towards the outside and inside of the membrane. Let's say this is the inside of the cell, this is the outside, and the fatty acids in the middle of the membrane. Lipids, in general, are what you would call hydrophobic. That is, they do not have a charge, so they don't stick to water. They do not dissolve in water. So fats and oils, there is no charge in this molecule, so they do not dissolve in water, as we know. Now, when a phospholipid, though, does have on the phosphate end, it has a charge. And so that end of the molecule will stick to water. And so when you take phospholipids and form this bilayer, they naturally form with the phosphate on the outside and the fatty acids on the inside. The phospholipid bilayer. All right, another kind of lipid, which is sort of completely different from the others, is known as a steroid. And we saw that in the previous chapter with the estrogens and testosterone. And here's that basic structure. You can see this one is different from those It's cholesterol. Um, and cholesterol is a necessary component in our diet, although you don't want too much of it, but because cholesterol is also found in cell membranes. You get, again, you got your phospholipids here, but you have cholesterol stuck in. And what the cholesterols do for the cell membrane is make it more fluid. So you can have more cholesterols, which make the membrane even more flexible and fluid, or fewer cholesterols, and it becomes more rigid. Um, and this level can be adjusted as needed. Um, cholesterol can be found in the diet. Um, I remember from the reading, I think it's also synthesized in the liver. Um, yeah, and it's the precursor to making other types of steroids. So this cholesterol that you consume and make in your body can be turned into other types of steroids like testosterone or estrogens or also all sorts of types of steroids. Okay, so those are the lipids. Um, I'm a little short on time. I'm going to break this chapter up into two, so I'll stop here and make a separate video for sections 4 and 5.